Okay, very good evening, everybody. Today we're going to talk about the second portion of your biochemistry. So, on the live session, so let me open the things for you. Yesterday we have talked till now, we have talked so far your till your lipoproteins we have talked so far. So now speaking about it. Now speaking about the next portion, which is your fatty acid. Okay, so we will do the remaining biochemistry today, and we will do anesthesia today. So we are having two things on the agenda today. One is your remaining portion of your biochemistry, which is your lipids, protein, amino acid, and vitamins and molecular biology. And second area of interest is your anesthesia. So speaking about the biochemistry is one of the subject where you get a pretty easy score. Like, you can score 17 out of 17. Out of 17, you may get around like 15 questions right if you know the concept very clearly. Okay, so just a very small things. But all of them are factual that you have to remember most of them. If you know that, then the biochemistry will be the piece of cake for you. Okay, so our notes is kind of like a very high yield one, where you may expect to see at least 10 to 12 questions out of 17 in biochemistry. So make sure you know and make sure you follow the PDF. Okay. So as I said, you have to integrate the PDF with your own notes, and you have to add the additional points, and then you have to compile it like your own notes. So this is how you have to do. So now speaking about it, number one is your fatty acid. So today the first topic we are going to talk about is your fatty acids. So we can talk about the fatty acids, both synthesis as well as your degradation. Speaking about the fatty acid synthesis, there are no, talking about the step-by-step pattern, it won't work for the fatty acid synthesis. So we have those important highlights that you have to remember for the fatty acid synthesis. What are the highlights you have to remember here in terms of fatty acid synthesis? Number one is your rate limiting enzyme, which is your acetyl-CoA carboxylase. Okay, rate limiting enzyme, which is your acetyl-CoA carboxylase, where it is stimulated by citrate and inhibited by fatty acyl-CoA. Very, 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 very important MCQ. And fatty acid synthesis requires NADP. Fatty acid synthesis requires NADP. Another important MCQ. So here 1 and 2 is your regulation, 3 is your rate limiting enzyme. All three are pretty important, people. Okay, then apart from that, one time in FMGE, they asked what are the components of your fast complex. All of the following are the components of your fast complex, except to ask the question. So speaking about the components of your fast complex, so one is your thioesterase, enoyl reductase, hydratase, ketoacyl synthase, ketoacyl reductase, and malonyl acetyl transferase. Malonyl or acetyl transferase are the 6 enzymes which belongs to your fatty acid synthase complex. So they may ask you as a question of a negation, all of the following are the components of your fast complex, except would be the question. So thioesterase, enoyl reductase, hydratase, ketoacyl synthase, ketoacyl reductase, and malonyl or acetyl transferase. Malonyl or acetyl transferase is the one. Then you have a degradation of your fatty acids. Speaking about the degradation, the important things is a degradation part. Understand it very carefully. This is kind of like we compile the high yield area where where you need to focus and where not you need to focus. It was been clearly written. Okay, make sure you know every single point of it. Okay, so first one is your rate limiting enzyme. Rate limiting enzyme is a carnitine acyl transferase 1. Rate limiting enzyme is a carnitine acyl transferase 1, and among that, carnitine is a membrane protein. Carnitine is a membrane protein which transports your acyl-CoA, which transports your acyl-CoA, carnitine is a membrane protein which transports your acyl-CoA from the cytosol to the mitochondria for your fatty acid degradation, okay, or beta oxidation of fatty acid. Okay, where does the location? What is the location of your degradation of fatty acid? For a very long chain and long chain fatty acid, it occurs in peroxisomes, and then it will be converted into medium chain fatty acid, and then it will goes into the mitochondrial matrix for further oxidation. So peroxisome will degrade it into medium chain fatty acid, and then medium chain fatty acid will be going under the further beta hydroxide. We have the beta oxidation proper in the mitochondria. And then short chain fatty acid will not undergo beta oxidation. Instead, it will undergo lysosomal hydrolysis. Lysosomal hydrolysis. So what are the things you have to remember? As I said, each and every point are important. One is your rate limiting enzyme, which is your carnitine acyl transferase 1, CAT1 or CPT1, carnitine palmitoyl transferase 1, any of the options they may give you. CAT1 or CPT1 is a rate limiting enzyme. Among the carnitine is a membrane protein which transport the acyl-CoA from cytosol to mitochondria. And what is the location of your fatty acid degradation? Very long chain fatty acid and long chain fatty acid, it is peroxisomes. Medium chain, it is mitochondria. And short chain, it is lysosomal hydrolysis. Okay. If they are asking you in general, where does the beta oxidation of fatty acid is taking place? And they are not giving you any particular area that it is they are not giving you whether it is a long chain fatty acid or a medium chain fatty acid, then your option is going to be your mitochondria. So if they ask you where does the location in the location of a beta oxidation of fatty acid, then the main option is your mitochondria. Okay, then you have the regulation. Carnitine shuttle is inhibited by melanoyl-CoA. Medium chain acyl-CoA dehydrogenase deficiency causes Jamaican vomiting sickness or sudden infant death syndrome. Jamaican vomiting sickness is due to consumption of your raw ackee fruit. Consumption of your raw ackee fruit. Raw ackee fruit, which contains a toxin called as hypoglycin A. Hypoglycin A. This toxin can able to break down or lose your medium chain acyl-CoA dehydrogenase. Will lose your medium chain acyl-CoA dehydrogenase or SIDS. The deficiency, congenital deficiency may gives rise to sudden infant death syndrome. Then you have this peroxisomal catalase deficiency. So peroxisomal catalase deficiency could cause a Zellweger syndrome. Peroxisomal catalase deficiency could cause a Zellweger syndrome, and absence of phytanic acid oxidase could cause Repsom's disease. Absence of phytanic acid oxidase could cause Repsom's disease. And what is the end product? For an end product, you have to remember it like a odd chain fatty acid. The end product is propenyl-CoA plus acetyl-CoA. Because one time in FMGE, they asked this as a question. The end product of odd chain fatty acid is dash. The options given were acetyl-CoA plus acetyl-CoA, propenyl-CoA plus acetyl-CoA, and propenyl-CoA plus propenyl-CoA, and propenyl-CoA plus butyryl-CoA. This was the option given to you. That time, most people are confused and they only knew the propenyl-CoA. So you have to know it's the odd chain fatty acid. The end product is propenyl-CoA plus acetyl-CoA. While even chain fatty acid, the end product is acetyl-CoA plus acetyl-CoA. So this is all about the fatty acid synthesis and degradation you have to know. So what are the things we are going to talk about here? So one is your synthesis of fatty acid. Synthesis requires NADP, first important highlighted MCQ. And the second rate limiting step is your acetyl-CoA carboxylase, another important highlighted MCQ. And it is stimulated by citrate and inhibited by your fatty acyl-CoA, another important MCQ. And the components of your fast complex, thioesterase, enoyl reductase, hydratase, ketoacyl synthase, ketoacyl reductase, melanoyl or acetyl transferase. Melanoyl or acetyl transferase are the components of your fast complex. Then degradation, rate limiting enzyme is your carnitine acyl transferase 1 or CAT1 or CPT1, while the membrane protein which transmits acyl-CoA is your carnitine, and location, very long chain peroxisome, and medium chain mitochondria, short chain lysosomal hydrolysis. And regulation, carnitine shuttle is inhibited by melanoyl-CoA. and impairment of medium chain acyl-CoA dehydrogenase could result in either due to the consumption of ackee fruit, which results in Jamaican vomiting sickness, or it may be due to the SIDS, sudden infant death syndrome. Okay. Then you have the peroxisomal catalase deficiency could result in Zellweger syndrome, or phytanic acid oxidase deficiency could result in Refsum's disease. So the Refsum's disease, sometimes they ask as a clinical features. So the clinical features of your Refsum's disease are cerebellar ataxia, and peripheral neuropathy. You have to remember the clinical feature. Cerebellar ataxia and peripheral neuropathy. Then you have a cholesterol synthesis. Rate limiting enzyme is your HMG-CoA reductase, inhibited by statins. First formed steroid is a lanosterol. These are the three words you have to remember. Almost 17 to 18 FMGE, they had asked this question. So you may not expect, but luckily if you expect, you can answer. Only one line in cholesterol synthesis you have to remember is the rate limiting enzyme of cholesterol synthesis is HMG-CoA reductase, which is inhibited by the drugs with the suffix of statins. And the first formed steroid is lanosterol. The first formed steroid is your lanosterol. Then you have a ketone bodies. Speaking about the ketone bodies synthesis occurs in starvation, and a test for your ketone bodies is Rothera's test. And on a prolonged starvation, your brain, heart, and skeletal muscle uses ketone bodies as a source of energy. And you have to remember that the rate limiting enzyme of your ketone body synthesis is HMG-CoA synthase. HMG-CoA synthase. Can you able to see here, people? HMG-CoA synthase. So what is the rate limiting enzyme of your ketone body synthesis, which is your HMG-CoA synthase. Okay, so far we have covered the lipid. So shall we revise the lipid, people? Faster. Okay. Now speaking about the lipid, the first thing, what are omega-3 fatty acid? Omega-3 fatty acid, what are they? What are the omega-3 fatty acid? Cervonic acid, timnodonic acid, and alpha-linolenic acid. Okay. Among that, cervonic acid is also known as docosahexaenoic acid, which is present in breast milk. Then you have omega-6 fatty acid, which is your gamma-linolenic acid, linoleic acid, and then arachidonic acid, which is a semi-essential fatty acid, is the arachidonic acid, and arachidonic acid is responsible for the synthesis of prostaglandin. Very, 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 very important MCQ. Okay. Then you have a lipid storage diseases. Speaking about the lipid storage diseases, we know that there are certain few things. Number one is your sphingomyelinase enzyme. Sphingomyelinase enzyme defect could result in Niemann-Pick's disease, ceramidase, Farber's disease, and you have this glucocerebrosidase enzyme defect, Gaucher's, alpha galactocerebrosidase, Fabry's, and beta galactocerebrosidase, Krabbe's, hexosaminidase A def, Tay-Sachs, hexosaminidase A or B def, that is your Sandhoff's disease. Among that, Erlenmeyer flask deformity is seen in Gaucher's, wrinkled paper appearance is seen in Gaucher's, cherry red spot on macula is seen in Niemann-Pick's, mainly Niemann-Pick's. Tigroid appearance is seen in metachromatic leukodystrophy. Okay, metachromatic leukodystrophy. Function of your apoprotein B48 is to transfer the chylomicron from the small intestine. Deficiency could result in A-beta lipoproteinemia. Function of ApoC2 is activation of lipoprotein lipase. Deficiency result in type 1 hyperlipoproteinemia. Apart from that, you have a ApoE, which mediates the receptor lipoprotein interaction. Deficiency could result in type 3 dysbeta lipoproteinemia or remnant removal disease or broad beta disease. Okay, then you have a ApoA1. Main function of ApoA1 is it is present in HDL. It activates LCAT, lecithin cholesterol acyl transferase. Even once they asked in FMGE the question of expand LCAT, people, you have to remember lecithin cholesterol acyl transferase. Let me write it on the side. You can able to understand that too. Lecithin cholesterol acyltransferase. The very closest option to confuse you was the acetyltransferase. Okay, so lecithin cholesterol acyltransferase is so called as LCAT. One time they asked this as a question. Expand LCAT was on once the MCQ. Okay, so you should know that. Okay, apart from that, you have the fatty acid synthesis. Rate limiting enzyme is your acetyl-CoA carboxylase, and it is required for what is required for the fatty acid synthesis? NADP, and a component of fast complex of thioesterase, enoyl reductase, hydratase, ketoacyl synthase, ketoacyl reductase, melanoyl or acetyl transferase. Melanoyl or acetyl transferase. Then degradation, rate limiting enzyme is CAT1. Location of very long chain fatty acid is peroxisome. Medium chain is mitochondria. Short chain is lysosomal hydrolysis. Peroxisomal catalase deficiency could result in Zellweger syndrome. Phytanic acid oxidase deficiency could result in Repsom's disease. End product of short chain fatty acid is propynyl-CoA plus acetyl-CoA, while end product of even chain fatty acid is your acetyl-CoA plus acetyl-CoA. And what is the cholesterol synthesis? Rate limiting enzyme is HMG-CoA reductase, which is inhibited by statins. And then first formed steroid in the cholesterol synthesis is lanosterol. Rate limiting enzyme of ketone body synthesis is HMG-CoA synthase. It is a ketone body synthesis mostly occurs in the starvation. So on prolonged starvation, your brain, heart, and skeletal muscle uses ketone bodies as a source of energy. And the test for ketone bodies is your Roth-Ras test. Test for your ketone bodies is your Roth-Ras test. So this is all about the lipid, people. So are you clear with the lipids? Shall I move on to the vitamins? Shall I move on to the vitamins? Shall I move on to the vitamins? Okay, speaking about the vitamins. So vitamins are classified into two types. So vitamins are classified into two types, namely fat-soluble and water-soluble vitamin. Namely fat-soluble and water-soluble vitamin. What are fat-soluble vitamin? A, D, E, and K are your fat-soluble vitamin. What are water-soluble one? B complex and vitamin C. B complex and vitamin C are water-soluble one. Okay, fine. Among the fat-soluble vitamin, we will discuss first one by one. The first one we are going to talk about is your vitamin A. The first one we are going to talk about is your vitamin A, where you see the structure of your vitamin A was well asked once on an MCQ as a beta adenine ring plus isoprenoid side chain. What was that? Beta adenine ring plus isoprenoid side chain. And what are the active form of vitamin A? The active form of vitamin A are retinol, retinol, and retinoic acid. Retinol, retinol, and retinoic acid. Okay, you have to remember this as a source. We can able to remember each and every vitamins like this, or maybe you can able to make a table like an active form, function, deficiency, or excess dose. Okay, these are the 5 areas we are going to talk about it. Okay, so first one is your active form of vitamin A, retinol, and retinoic acid. And the function, very important MCQ, people. The function of your fat soluble vitamin is conversion of 11-cis retinol to all trans retinol. Conversion of 11-cis retinol to all trans retinol. And the second most important function is your gene expression and differentiation. And it is also an antioxidant. And it is also responsible for the maintenance of your normal epithelium. Maintenance of your normal epithelium. Deficiency causes xerophthalmia and hyperkeratinization. Deficiency causes xerophthalmia and hyperkeratinization. If you are grading the WHO stages of your xerophthalmia, WHO staging of xerophthalmia, XN indicates night blindness. XN indicates night blindness. And X1B indicates Bitot spot. Very important MCQ, which is an ophthalmology MCQ, but I am covering it up here itself. Okay. Night blindness is XN and X1B is Bitot spot. Very important. And toxicity could result in benign intracranial hypertension or your pseudotumor cerebri. Then vitamin D. Active form of vitamin D is so called as 1,25 dihydroxycholecalciferol. So how does this vitamin D is synthesized? So the vitamin D, whenever the sunlight hits on your skin, your cholesterol will be converted into 7-dehydrocholesterol or cholecalciferol. It goes to liver, where your cholecalciferol will be converted into 25-hydroxycholecalciferol. It will be converted into 25-hydroxycholecalciferol, and then it goes to the kidney, where your 25-hydroxycholecalciferol will be converted into 1,25 dihydroxycholecalciferol mediated by 1-alpha hydroxylase enzyme. Mediated by 1-alpha hydroxylase enzyme. Okay, what is the main function? Main function is maintenance of your calcium homeostasis. Maintenance of your calcium homeostasis. While deficiency could result in rickets and osteomalacia. Deficiency could result in rickets and osteomalacia, where the clinical features, this is quite 5-star rated topic. If they want to ask you with the orthopedic or pediatric question, they may give you this one of the clinical features and they may ask you this. Okay, very, very important. So the clinical features of rickets are rachitic rosary, wind-swept deformity, people. Very, 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 very important MCQ. The classical sign of your rickets is your wind-swept deformity, where one side you see the valgum, another side you see the varum. One side you see the valgum, another side you see the varum. So this is how the body of the or the limbs of your individual will be there. You have those limbs of your individual. Lower limb considered. So this is your person, and you see there are two legs. Whenever the wind is sweeping him, his legs will move on to this direction. This type of deformity is so called as wind-swept deformity, which is seen in the rickets. Apart from that, you may see the Harrison groove, delayed dentition and delayed closure of fontanelle. And you have the vertebral column abnormalities like kyphosis, scoliosis, and lordosis, etc. All of them could be resulting in this condition, okay, due to the rickets. So we have talked about A and D. So vitamin A, structure is beta-ion ring plus isoprenoid side chain. Active form is retinol, retinol, and retinoic acid. Main function is conversion of 11-cis retinol to all-trans retinol. Deficiency is xerophthalmia and hyperkeratinization. Toxicity could result in pseudotumor cerebri. While vitamin D, active form is 1,25 dihydroxycholecalciferol. Synthesis from sunlight, cholesterol to cholecalciferol. Cholecalciferol goes to liver, 25 hydroxycholecalciferol, and then it goes to kidney, where 1,25 dihydroxycholecalciferol. Then function, maintenance of calcium homeostasis, deficiency, rickets, and osteomalacia, where you see the rachitic rosary, wind-swept deformity, Harrison groove, and delayed dentition, kyphosis, scoliosis, and lordosis. Next is a E. E, only 1 point. Active form of vitamin E is tocopherol. So tocopherol binds with the accidents and forms a tocopherol radical. So it will bind with the free radical and convert the free radical to tocopherol radical and eliminate them. So deficiency is quite rare, but it can cause us a hemolytic anemia and spinocerebellar ataxia. Quite a rare one. And vitamin E, so far vitamin E, the only point you have to know is active form is alpha tocopherol. It binds with the free radical and converts the free radicals to tocopherol radical and eliminate them. That's why it is an antioxidant. It is an antioxidant. Then vitamin K, active form is K1, phylloquinone, menaquinone, and menadione. One time this was asked as a FMGE question. Okay, one time this was asked as a FMGE question. What is vitamin K2? The options were phylloquinone, menaquinone, menadione, and phyllodione. So you have to choose which one is a K2. Menaquinone is the answer. Okay, so remember active form is K1, it is phylloquinone, K2, it is menaquinone, and K3, it is menadione. K3, it is menadione. And what are the functions? Two important function, people, that you should not miss. Very, 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 very important MCQ again. It was asked in the previous MCA exams as well. One is your gamma carboxylation of glutamic acid. Gamma carboxylation of glutamic acid, because once in FMG they asked main function of your vitamin K is dash, and in the option they had given was gamma carboxylation of glutamic acid, gamma hydroxylation of glutamic acid, gamma carboxylation of aspartic acid, and gamma hydroxylation of aspartic acid was a question. So you should know it is a gamma carboxylation of glutamic acid. Both carboxylation as well as glutamic acid you have to remember. Then you have to remember vitamin K dependent clotting factors. I used to remember with the mnemonic of company started in 1972. Company started in 1972. So 2, 7, 9, 10, 2, 7, 9, 10, protein C and protein S. 2, 7, 9, 10, protein C and protein S was the mnemonic factor. 2, 7, 9, 10, protein C and protein S. And deficiency causes hemorrhagic diseases in newborn. Deficiency causes hemorrhagic diseases in the newborn. And toxicity causes hemolysis, kernicterus, hyperbilirubinemia, and brain damage. Kernicterus, hyperbilirubinemia, and brain damage. While the antidote is, remember always, warfarin and vitamin K are like brothers. So warfarin counteract vitamin K. Vitamin K counteracts warfarin. Okay, vitamin K counteracts warfarin, and warfarin counteracts vitamin K. Antidote for vitamin K toxicity is warfarin, while antidote for warfarin toxicity is vitamin K. You have to remember that. Okay, this is all about the fat soluble vitamin people. So vitamin A, beta iron ring plus isoprenoid side chain is a structure. Active form, retinol, retinoic acid. Function, conversion of 11 cis retinol to all trans retinol. Deficiency is ophthalmia and hyperkeratinization. Toxicity, pseudotumor cerebri. Vitamin D, active form is 1,25 dihydroxycholecalciferol. Synthesis, cholesterol to 7 dehydrocholesterol. 25 hydroxycholecalciferol. 1,25 dihydroxycholecalciferol in kidney. Function, calcium homeostasis. Deficiency, rickets and osteomalacia. Vitamin E, tocopherol. Active form is tocopherol. Function is antioxidant by forming a tocopherol radical. And then vitamin K, active form. K1 is phylloquinone. K2 is menaquinone. K3 is menadione. Function, gamma carboxylation of glutamic acid. Clotting cascade factor 2, 7, 9, 10, protein C and protein S. Deficiency, hemorrhagic diseases in newborn, while antidote, it is a warfarin. Okay, hemorrhagic diseases in newborn, while it is an antidote, it is a warfarin. Okay, next, we move on to water-soluble vitamins. Vitamin B1, active form is thymine pyrophosphate TPP. As we studied in the PDH complex, TPP is required in. TPP is a component of tender loving care for Nancy. Yes or no? Which is required in three enzyme complexes. What are they? It is required in PDH complex, alpha ketoglutarate dehydrogenase complex, PDH complex, alpha ketoglutarate dehydrogenase complex, branched chain keto acid dehydrogenase complex. Branched chain keto acid dehydrogenase complex. So all of them will be requiring a thymine pyrophosphate as a cofactor. Now, deficiency causes beriberi. So, speaking about the vitamin B1, any of these two options will be given as a clue if they are giving you a case-based question. One, there will be either a history of alcoholism. The patient may be chronic alcoholic. The classical vitamin D, vitamin deficiency seen in this patient is B1. Or they may give you the consumption of polished rice. But this is quite rare. But maybe alcoholic history, there are a lot of questions in FMGE nowadays. They are coming with the vitamin B1 and history of alcoholism. So you should remember that thing. And how do you determine the how do you determine the vitamin B1 assay, which is a RBC transketolase, because transketolase requires TPP as a cofactor, which is an enzyme of HMP shunt. Okay, so RBC transketolase requires this. Okay, and apart from that, very dry and wet. Dry is associated with dry, yes or no? So neurological symptoms, while wet, it is associated with the cardiovascular symptom. Wet is associated with the cardiovascular symptom. And then we have vitamin B2, also known as Warnberg yellow enzyme. Active form is FAD and FMN. Deficiency causes ariboflavinosis, where you see this thing. Anywhere you see cheiliosis, angular stomatitis, or magenta colored tongue, or any mouth ulcer related to the corner of the lips or mouth, then immediately choose the vitamin deficiency is always be a, the vitamin deficiency you may see will always be a vitamin B2 deficiency. Vitamin B2 deficiency. Then you have vitamin B3, which is synthesized from tryptophan, and active form is NAD and NADP. Active form is NAD and NADP, while deficiency pellagra, 4 Ds of pellagra, diarrhea, dermatitis, dementia, and death. Among the dermatitis will be in the Casal's necklace pattern. Dermatitis will be in the Casal's necklace pattern. You have to remember that. So Casal's necklace is due to vitamin B3 deficiency. Toxicity causes cutaneous flushing and fulminant hepatitis. Very, very important MCQ. Okay, very, very important MCQ. Then you have vitamin B5. Active form is coenzyme A. Deficiency causes Gopalan's burning food syndrome. Deficiency causes Gopalan's burning food syndrome. And vitamin B6, active form is PLP. Function, it is required in most of the reaction. And assay for the PLP is RBC transaminase and high dose pyridoxine. This is MCQ, people. High dose pyridoxine is used for treatment of carpal tunnel syndrome, premenstrual syndrome, schizophrenia, and diabetic neuropathy. Carpal tunnel, premenstrual, schizophrenia, and diabetic neuropathy, where you see the high dose pyridoxal phosphate usage. Very, 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 very important MCQ, especially the carpal tunnel one. Especially the carpal tunnel one. Then vitamin B7, function is carboxylase reaction. So remember always, whenever you see the enzyme carboxylase, you see the cofactor is biotin. Blindly choose. Whenever you see the enzyme as a carboxylase, enzyme ends with a carboxylase, then the cofactor is always biotin. You can blindly choose. And deficiency is very, very rare, but deficiency can be associated with the consumption of raw egg white. Consumption of raw egg white, which contains a protein called as avidin, and this avidin is an anti-egg white factor. So that's why vitamin B7 or biotin is also known as anti-egg white injury factor. Anti-egg white injury factor. Then folic acid, location of absorption is jejunum and participate in 1-carbon metabolism. Participates in 1-carbon metabolism. Then vitamin B12, location of absorption is terminal ileum. It requires cobalt as a cofactor. And deficiency could result in megaloblastic anemia and subacute combined degeneration of spinal cord. Subacute combined degeneration of spinal cord or megaloblastic anemia. You have to remember that. Okay, you have to remember these things. And apart from that, vitamin C, active form is L-ascorbic acid. Active form is L-ascorbic acid. It is an antioxidant one used for collagen synthesis. Very, 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 very important. Bile acid synthesis, another important. And tyrosine metabolism, another important. These three are pretty important that you should have to remember that. Vitamin C is required for antioxidant, collagen synthesis, bile acid synthesis, and tyrosine metabolism. And deficiency causes scurvy. So you see the scorbutic rosary, and you see anywhere bleeding, like a bleeding in the gums or anywhere in the body, like it will be showing as a ecchymosis or petechiae or any splinter hemorrhage. All of these things can be resulting in the scurvy. Okay, all of these things can be resulting in the scurvy. So this is all about the vitamins, people. Now, moving on to the minerals, important minerals and their deficiency. Selenium deficiency could result in Keshan's disease. Copper deficiency could result in Menkes. Fluoride 1, dental caries. Cobalt 1, pernicious anemia. Iron 1, microcytic hypochromic anemia. Potassium, cardiac arrhythmia. Zinc, you may see the poor wound healing and growth retardation. Dermatitis. Okay, very, 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 very important is dermatitis, hypogonadism, and infertility. Dermatitis, hypogonadism, and infertility are due to the zinc deficiency. Okay, so these are the important deficiencies and associated diseases. So far we have completed carbohydrate, and we have completed lipids, and we have completed vitamins and minerals. So the next area of interest is the miscellaneous one. Then we move on to the protein and amino acid. Is the miscellaneous ones, but we move on to the protein and amino acids here. So speaking about that, first miscellaneous topic is your heme synthesis. Very, very important nowadays, because a lot of questions are being asked through this chapter. So you have to remember this heme synthesis. So speaking about the heme synthesis, the one which we are having is your succinyl-CoA and glycine. Succinyl-CoA and glycine, where you see the succinyl-CoA combines with the glycine and converts into delta ALA, where you see the delta ALA synthase, And apart from the delta ALA will be converted into 4,4-bilinogen people. Delta ALA will be converted into 4,4-bilinogen mediated by delta ALA dehydratase. Which is inhibited in lead poisoning. So whenever you see the vignette with the history of paint, exposure to paint, or the house has been painted recently, this type of a clue they may give you for the lead poisoning. It inhibits the delta ALA dehydratase, which prevents the conversion of delta ALA to 4,4-bilinogen. And then from 4,4-bilinogen to hydroxymethylbilane. 4,4-bilinogen to hydroxymethylbilane mediated by the enzyme hydroxymethylbilane synthase. Deficiency causes acute intermittent porphyria. And then you have this uroporphyrinogen 3. Conversion of HMB to uroporphyrinogen 3 is mediated by uroporphyrinogen 3 synthase, where you will be deficiency causes congenital erythropoietic porphyria. Then uro to copro mediated by urodecarboxylase, where the enzyme defect is porphyria cutaneous tarda. Coproporphyrinogen to protoporphyrinogen 3 mediated by coprooxidase, where the deficiency could result in hereditary coproporphyria. And protoporphyrinogen 3 to protoporphyrin mediated by protoporphyrinogen oxidase, where deficiency results in variegate porphyria. And protoporphyrin to heme mediated by ferrochelatase, where the enzyme deficiency could result in protoporphyria or erythropoietic protoporphyria. Okay, so far. So how do you remember it? Succinyl-CoA plus glycine gives rise to delta ALA synthase, which is a rate-limiting enzyme. Delta ALA to 4,4-bilinogen. 4,4-bilinogen to HMB. HMB conversion is mediated by HMB synthase. Deficiency causes AIP, acute intermittent porphyria. HMB to uro mediated by urosynthase, congenital erythropoietic porphyria. Uro to copro mediated by urodecarboxylase, deficiency, porphyria cutaneous tarda, which is PCT. And copro to proto mediated by coprooxidase, hereditary coproporphyria. Proto to protoporphyrin oxidase, variegate porphyria. And proto to heme, ferrochelatase, congenital erythropoietic protoporphyria. Then the test for 4,4-bilinogen is your Eldridge aldehyde test. Eldridge aldehyde test is the test we use for your heme synthesis. We use this for your testing of 4,4-bilinogen, specifically speaking, 4,4-bilinogen. Again, nowadays it has become like a 5-star rated topic because they had asked so much of questions from all the areas of your biochemistry. So now your heme synthesis is considered to be one of the topic of interest where you may expect most and most of the MCQs. So make sure you know the heme synthesis pathway. Next to the heme synthesis, the second high yield area is your collagen synthesis. Next to the heme synthesis, the second high yield area is your collagen synthesis. Speaking about the collagen synthesis, we have the pre-pro collagen. Pre-pro collagen contain glycine with another 2 amino acid. Most probable amino acid could be proline and lysine. And the pre-pro collagen will be converted into, pre-pro collagen will undergo a lot of sequence to form a pro collagen. One is a hydroxylation. So pre-pro collagen undergoes hydroxylation followed by glycosylation. glycosylation to procollagen, procollagen to trophocollagen, trophocollagen to normal collagen. Okay, trophocollagen to normal collagen. So procollagen is formation of procollagen is a formation of triple helix one. So triple helix formation is impaired in osteogenesis imperfectae. Triple helix formation is impaired in osteogenesis imperfectae. And conversion of pro to tropho is impaired in Ehlers-Danlos syndrome. Ehlers-Danlos syndrome. And conversion of tropho to collagen is impaired in Menkes disease. Is impaired in Menkes disease. Then you have a types of collagen and their location. Type 1, bone, and type 2, it is cartilage and nucleus pulposus. Type 3, it is blood vessels. Type 4, it is a basement membrane. Type 4, it is a basement membrane. So all of them will be moving over here so that you can able to understand. Apart from that, you have a protein and amino acid. What are essential amino acids? However, you used to remember, you can remember. Okay, some people used to remember it as like a Villambatti, and some people remember it as like a PVT Tim Hall. But however you want to remember, you can remember in that way. Okay, so the very classical essential amino acids are valine, isoleucine, leucine, lysine, arginine, methionine, phenylalanine, threonine, and tryptophan. Okay, threonine and tryptophan. Among that, arginine is a semi-essential amino acid. Arginine is a semi-essential amino acid where the part can be synthesized in your body. Okay, very, very important MCQ. Okay, then. So we classify the amino acid based upon the side chain. So first one is your aliphatic amino acid. Now speaking about the aliphatic amino acid, the very first one we asked to remember is, I used to remember the aliphatic amino acids as I love very attractive girls. I love very attractive girls was the mnemonic. Okay, I love very attractive girls was the mnemonic. I stands for isoleucine, leucine, leucine, valine, alanine, and glycine. Alanine and glycine. Okay, isoleucine, leucine, valine, alanine, and glycine are the area where you see this. And glycine is responsible for synthesis of three substances. One is your formation of glutathione, formation of creatinine, and heme synthesis. Formation of glutathione, formation of creatinine, and heme synthesis. And the synthesis of your aliphatic amino acids are from four sources. What are the four sources? Threonine via threonine aldolase, carbon dioxide and ammonia via glycine synthase, serine, and glyoxylate via glycine transaminase. Glyoxylate via glycine transaminase. And the primary hyperoxaluria type 1 is due to the deficiency of this glycine transaminase. Very, 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 very important MCQ. Okay, primary hyperoxaluria is a classical hyperoxaluria, which is due to the enzyme defect of glycine transaminase. So what are the things you have to know about the glycine? Formation of glutathione, formation of creatinine, and heme synthesis. Synthesis involves threonine, carbon dioxide and ammonia, serine, and glyoxylate. Primary hyperoxaluria type 1 is due to deficiency is due to glycine transaminase. Glycine transaminase. Then you have the alanine. Alanine is a non-essential amino acid that can be synthesized in your body from the glucose. And alanine is the most glucogenic and non-polar amino acid. And alanine can be synthesized from your, glucose can be synthesized from your body via Cahill cycle. So you have to know the alanine already. Now is your branched chain amino acid. What are the branched chain amino acid? We used to remember it with the mnemonic of I love Valeria. Okay, I love Valeria. So isoleucine, leucine, and valine are the branched chain amino acid. Branched chain aliphatic amino acids are ILV. Okay, branched chain aliphatic amino acids are ILV. That is your isoleucine, leucine, and valine. So speaking about that, we keep the isoleucine and valine together and leucine as separate. Speaking about that, we keep isoleucine and valine as together and then leucine as separate, and they can be converted into their respective acyl CoA, and then to their respective CoA is like a isoleucine and valine can yield a propionyl CoA, while your leucine can yield a acetyl CoA. Your leucine can yield a acetyl CoA. So this entire conversion is happening in a many step. One important step is this: branched chain keto acid dehydrogenase or decarboxylase. Branched chain keto acid dehydrogenase or decarboxylase enzyme defect. Defect could result in this enzyme defect could result in maple syrup urine disease, where you see the burnt sugar order in the urine. You see the burnt sugar order in the urine. Okay, very, very important one. Then apart from that, there is a one intermediate complex, isovaleric CoA dehydrogenase. Deficiency causes isovaleric acidemia, where you see the sweaty feet order. Where you see the sweaty feet order. So burnt sugar order, maple syrup urine disease. Enzyme defect, branched chain keto acid dehydrogenase or decarboxylase. Then you have this isovaleric CoA dehydrogenase defect, isovaleric acidemia, while the enzyme defect is, what type of urine order you may see? Sweaty feet order you may see. What type of urine order you may see? Sweaty feet order you may see. Then apart from that, aromatic amino acid. Aromatic amino acid, I used to remember with the mnemonic of PTT, phenylalanine, tyrosine, and tryptophan. Phenylalanine, tyrosine, and tryptophan. So speaking about the phenylalanine to tyrosine conversion is mediated by phenylalanine hydroxylase, which requires tetrahydrobiopterin as a cofactor. This tetrahydrobiopterin is also required in tyrosine to dopa conversion and tryptophan to 5-hydroxytryptophan conversion and arginine to citrulline conversion. So all of them will be requiring this tetrahydrobiopterin. One such important one is it is also required in phenylalanine hydroxylase, and deficiency of this phenylalanine hydroxylase could result in phenylketonuria. Where you see the mousy odor or musty odor in urine. Okay, mousy odor or musty odor in the urine. Okay, apart from the light skin, blue eyes, mousy odor or musty odor in the urine indicates it is a phenylketonuria. And the test for the phenylketonuria screening is Guthries and confirmatory is tandem mass spectrometry. Okay, screening is Guthries and confirmatory is tandem mass spectrometry. And the ketone for a phenyl pyruvate and a phenyl acetate, which has been excreted in the urine, that's why you get a phenylketonuria. Among that, phenyl pyruvate is determined or diagnosed by ferric chloride test. Very, 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 very important MCQ. This ferric chloride test is the test where you used to diagnose the phenyl pyruvate. Okay. Now, apart from the tyrosine, synthesis of catecholamines, thyroid hormones, and melanin. Melanin via tyrosinase deficiency causes albinism. And catecholamine, dopamine, epinephrine, norepinephrine will be there, and thyroid hormones has been synthesized. Synthesis of thyroid hormones has been discussed already in the physiology, and catecholamines we will discuss that in the ANS pharmacology. So clinical correlation, two things in the tyrosine: alkaptonuria, absence of homogentisate oxidase enzyme, where you see the dark color urine or Coca-Cola colored urine. Very, 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 very important MCQ. Very, 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 very important MCQ. So alkaptonuria is due to the homogentisate oxidase deficiency, and you see the dark color urine. And dark colored pigmentation on the skin. The dark colored pigmentation on the skin is so called as ochronosis. The dark colored pigmentation of the skin is so called as ochronosis. Then you have a tyrosinemia. Tyrosinemia types are type 1, type 2, and type 3. Type 1, type 2, and type 3. Type 1 is due to the fumaryl acetoacetate hydrolase. where you see the boiled cabbage odor. Type 2 is tyrosine transaminase. And type 3 is para-hydroxyphenylpyruvate hydroxylase deficiency. Okay, para-hydroxyphenylpyruvate hydroxylase deficiency. So we have talked about the aromatic amino acid phenylalanine and tyrosine. The last one is your tryptophan. Synthesis of three things: serotonin, melatonin, niacin. Serotonin, melatonin, niacin. Among that, the chemical name of your serotonin is 5-hydroxytryptamine. Remember, most people used to confuse it with the 5-hydroxytryptophan. No, it is not 5-hydroxytryptophan. It is a 5-hydroxytryptamine. Remember, don't confuse. Okay, then you have the niacin. And deficiency could result in Hartnup disease and carcinoid syndrome. Hartnup disease is due to defect in neutral amino acid transporter, while carcinoid, it is due to the excessive serotonin producing tumor, where you see the elevated hydroxyindoleacetic acid level. Elevated hydroxyindoleacetic acid levels, you may see it in the carcinoid syndrome. Okay, carcinoid syndrome. So this is all about the aromatic amino acids, people. This is all about the aromatic amino acids. That is a PTT. Apart from that, you have this arginine, lysine, and histidine. Arginine is responsible for the synthesis of nitric oxide by stimulation of guanylate cyclase, activation of GTP, and produces vasodilation and augmentin. Very, very important MCQ. Apart from that, lysine, synthesis of collagen, and carnitine along with the methionine. Another important MCQ. Histidine, synthesis of histamine, anserin, carnosine, and ergothioneine. Histamine, anserin, carnosine, and ergothioneine is a histidine. Okay. Apart from that, you have a sulfur-containing amino acid, where you see the methionine and the cysteine. Homocystinuria is due to there are only two important conditions. One is your homocystinuria, another one is your Osterhaus syndrome. Homocystinuria is due to the cystathione synthase defect, where you see the ectopia lentis or subluxation of lens, mental retardation, Marfan-like habitus, and thromboembolism. Marfan-like habitus and thromboembolism. While in the case of your Osterhaus syndrome, where you see the malabsorption of methionine and other neutral amino acids. Malabsorption of methionine and other neutral amino acid. Then you have the urea cycle. So speaking about the urea cycle, I rather call it as 5 enzymes, 5 defects. 5 enzymes, 5 diseases. You have to remember. So first we will discuss about that. So the first enzyme is ammonia plus bicarbonate plus 2 ATP combined to form carbamyl phosphate mediated by carbamyl phosphate synthase 1. Mediated by carbamyl phosphate synthase 1, where deficiency could result in hyperammonemia type 1. Deficiency could result in hyperammonemia type 1. While the carbamyl phosphate will combine with the ornithine and converts into citrulline mediated by the enzyme OTC, that is ornithine transcarbamylase. Mediated by the enzyme OTC, that is your ornithine transcarbamylase. Deficiency could result in hyperammonemia type 2. Hyperammonemia type 2. Then the formed citrulline will combine with the aspartate and form argininosuccinate mediated by argininosuccinate synthase. Deficiency could result in citrullinemia type 1. And argininosuccinate will be broken down into arginine and eliminates fumarate mediated by argininosuccinate lyase, where the enzyme defect could result in argininosuccinic aciduria. And then you have the arginine will be converted into ornithine by arginase, while the enzyme defect is argininemia. While the enzyme defect is argininemia. Okay, so 5 enzymes, 5 diseases. So 5 enzymes are CPS1, OTC, argininosuccinate synthase, argininosuccinate lyase, and arginase. What are the diseases? So CPS1 defect could result in hyperammonemia type 1. OTC defect could result in hyperammonemia type 2, and argininosuccinate synthase defect could result in citrullinemia type 1. Argininosuccinate lyase defect could result in argininosuccinic aciduria. Arginase deficiency could result in argininemia. Now, coming to the FMGE highlight points. What are the things the FMGE may test you with the urea cycle? So, first point is, what is the location of urea cycle? What is the location of urea cycle? One is your liver. In the organ, it is a liver. Organelle, it is both cytosol and mitochondria. Both cytosol and mitochondria. We studied in the gluconeogenesis that hug requires both. H stands for heme synthesis, U stands for urea cycle, G stands for gluconeogenesis. Among that, we studied all the three cycles now. Yes or no? All the three cycles now. Now, organelle is both cytosol and mitochondria. And apart from that, rate limiting enzyme is your CPS1. CPS1. Orotic aciduria is seen in. Orotic aciduria is seen in CPS, sorry, that is a hyperammonemia type 2 OTC defect. Hyperammonemia type 2, that is OTC defect. Enzyme defect is ornithine transcarbamylase, OTC enzyme defect. Enzyme defect, we can say. These are the thing you have to know. Okay. Then apart from the triple H syndrome, that is a triple H syndrome is due to ORNT1 gene defect, where you see the impaired transport of your ornithine. Impaired transport of your ornithine is your triple H syndrome. Then apart from that protein, so primary structure, linear chain of amino acid bounded by covalent bond, and secondary structure, folding of your one primary structure is so called as secondary, where it is containing a hydrogen bond. And remember two important facts, that alpha helix and beta plated sheet, alpha helix and beta plated sheet belongs to secondary structure. Very, 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 very important MCQ. Belongs to secondary structure of protein. While tertiary is single, one complete turn is tertiary, and more than one complete turn is so called as multiple turns, and multiple foldings is so called as quaternary. Multiple foldings and multiple turns is so called as quaternary. With this, we are completing our proteins and amino acids. With this, we are completing our proteins and amino acids. So far, we have done our carbohydrates, lipids, proteins, amino acids, and vitamins, and miscellaneous important topic. So the only topic left over is your electron transport chain, and then you have your molecular biology, and remaining leftovers are pending. So we will do that after the break. Kindly take a 10 minutes of break and see you all by 6:10. Take a 10 minutes of break and see you all by 6:10. Okay. So far we have done the so far we have done the amino acids, proteins, and vitamins, and lipids and carbohydrate. So the next area of interest is your electron transport chain and their complex inhibitor. So this table is kind of like a 7-star rated topic because nowadays the table has been asked so many times, so you should not miss it. So first one is your complex number 1, which is the NADH dehydrogenase, which is inhibited by rotenone, phenobarbital, pyrisidine, and amytal. Rotenone, phenobarbital, pyrisidine, and amytal. And you have this complex 2, where it is the enzyme complex name is succinate dehydrogenase and inhibited by your malonate. And then complex 3, which is the cytochrome oxidoreductase, and cytochrome oxidoreductase is inhibited by fenformin and antimycin A. And complex 4, which is the cytochrome oxidase, inhibited by carbon monoxide, cyanide, and H2S. Carbon monoxide, cyanide, and H2S. While complex 5 is ATP synthase, inhibited by oligomycin and attractyloside. And then you have the uncouplers. What are the uncouplers? We have the dinitroprenol, thyroxine, carboxin, oligomycin, rotamycin, aspirin, and thermogenin. Oligomycin, rotamycin, aspirin, and thermogenin. Among that, your tyrosine and thermogenin are the physiological uncoupler. Among that, your tyrosine and thermogenin are physiological uncoupler. You should remember that, people. Physiological uncouplers. Okay. So either they may ask you the name of the complex, or they may ask you like which one is oxygen dependent. Complex 4 is a oxygen dependent. Complex 4 is a O2 dependent one. Either they may ask you which one is oxygen dependent, or what is the name of complex 2 or 3 or 4 or 5, or they may ask you the complex inhibitor, or they may ask you the uncouplers. And now recently they asked the thyroxine, and they asked like thyroxine is a what type of uncoupler? It is a physiological uncoupler. Okay, so they are given a 4 options, and they asked which among the following is a physiological uncoupler, and the options were given as a tyrosine. You should not forget it. So electron transport chain table is kind of like a 7-star rated topic, people. You should not miss it. Okay. Then you have molecular biology. So speaking about the molecular biology, first one is nucleic acid. Nucleic acid is a sequence of nucleotide, and nucleotide composed of nucleoside plus phosphate. Nucleoside plus phosphate. And then nucleoside composed of sugar and nitrogenous base. Nucleoside composed of sugar and nitrogenous base. While sugar should be, in case of DNA, it is deoxyribose. In case of RNA, it is a ribose sugar. In case of RNA, it is a ribose sugar. And nitrogenous base could be of purines and pyrimidines. Purines were supposed to be adenine and guanine, while pyrimidines were supposed to be cytosine and thymine. Among that, purines are the 2-ringed structure. You have to remember that. Purines are the 2-ringed structure, and amino acid required for the synthesis of purines are aspartate, glutamate, and glycine. Amino acid required for the purine synthesis are aspartate, glutamate, and glycine. While in the case of pyrimidines are 1-ringed structure, and only glutamate and aspartate is required for the synthesis of pyrimidines. Only glutamate and aspartate is required for the synthesis of pyrimidine. And what is the rate limiting enzyme of pyrimidine synthesis? Which is a CPS2. And apart from that, you have a HGPRTase deficiency. HGPRTase deficiency could result in Lesch-Nyhan syndrome. HGPRTase deficiency could result in Lesch-Nyhan syndrome, where you see the self-mutilation and gout. So on the salvage pathway, we want to know that the end product is xanthine. Xanthine will be converted into uric acid, and uric acid will be excreted in the urine. So excessive uric acid may go and accumulate in the joint and can produce a gout, people. Excessive uric acid may go and accumulate in the joint and can produce a gout. So what is the treatment? The treatment is to inhibit this xanthine oxidase enzyme. Treatment is to inhibit this xanthine oxidase enzyme. So drugs such as allopurinol and febuxostat is used to inhibits the xanthine oxidase enzyme, which requires molybdenum as a cofactor. So whatever which has been given in this entire square, whatever which we have written in this entire square is a 7-star rated topic nowadays. Because in a 1 year back in a FMGE, they asked 3 questions within this box itself. So you should know that gout is also one of the topic of interest nowadays. Okay. So the end product of your salvage or the salvage pathway is xanthine. Xanthine will be converted into uric acid by the enzyme xanthine oxidase, which requires molybdenum as a cofactor, which is inhibited by your allopurinol or febuxostat, and this excessive uric acid may accumulate in the joint and produces the symptoms like gout. Okay, produces the symptoms like a gout. So these are the basics of a molecular biology, people. Now, what is the central dogma of the molecular biology? Which is the DNA to DNA is the replication, and the DNA synthesis of RNA from DNA is transcription, and synthesis of protein from RNA is translation. Synthesis of protein from RNA is translation. So speaking about the synthesis of DNA from DNA, which is the replication, where you see the unwinding of supercoiling is done by topoisomerase, and the strand breaking is done by helicase, and the prevention of reannealing is done by single-stranded binding protein, and leading strand elongation is done by DNA polymerase, and lagging strand elongation is done by trimase, DNA polymerase 1, and DNA ligase. So very, very important. Unwinding of supercoiling is done by topoisomerase, where drugs such as etoposide, teniposides are the topoisomerase inhibitor. Etoposide, teniposides are the topoisomerase inhibitor. Okay, and then you have the strand breaking, which is a helicase, and the prevention of reannealing is done by single-stranded binding protein, and leading strand elongation is done by DNA polymerase, while lagging strand elongation is done by primase, DNA polymerase 1, and DNA ligase. Primase, DNA polymerase 1, and DNA ligase. Okay, next is a mutation. Very, 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 very important is the mutation. One is your silent mutation, while the third base of the triplet is altered. So no change in amino acid. No change in amino acid synthesis. That's why it is so called as silent mutation. Missense mutation is first and second base of triplet is altered, so that there is a change in the amino acid. One such classical example is your sickle cell anemia, where your glutamic acid is replaced by valine. Yes or no? Sickle cell anemia is a type of missense mutation. Sickle cell anemia is a type of missense mutation. Then you have a nonsense mutation. I used to remember with the mnemonic of stop the nonsense. That is your early appearance of your stop codons. Early appearance of your stop codons, where you may see UGA, UAA, and UAG. UGA, UAA, and UAG are the nonsense mutation. Apart from that, frame shift mutation is nothing, just the addition or deletion of your nucleotide base in the sequence that lead to the formation of triplet codon. For example, consider we have the triplet codon of 1 to 7 base. For example, A, and this C, and G, T, A, G. Consider this is the sequence of your DNA sequence. Among that, if we remove that, for example, the triplets is going to be like this. Yes or no? If you remove the T out of it, your newly formed sequence of your triplet codon is AAA, CG, A, G. Yes or no? The forming amino acid is getting changed. Yes or no here? So that's why it is so called as frame shift mutation. That's why it is so called as frame shift mutation. And the second important thing is identification of palindromic sequence in the DNA. Identification of the palindromic sequence in the DNA. So palindromic sequence is nothing but the 3' to 5' direction. If they given you any sort of the 5' to 3' direction, if they given you any DNA sequence which are complementary to them, then that is so called as palindromic sequence. For example, A, T, and A, T, T. This is a palindromic sequence where the first one, if you are going in this, it will be a complementary to each other. If you are going like this, it will be complementary to each other. So a strand which contains both complementary strands, complementary nitrogenous base is so called as palindromic sequence in the DNA. Okay, you have to be remembering that. Okay, apart from that, there are types of DNA. Types of DNA, the most common form is your bDNA, which is a right-handed anti-parallel. What Watson and Crick had made it was a bDNA. What Watson and Crick was made, what Watson and Crick had made was a bDNA, while zDNA is a left-handed DNA. And there is a mitochondrial DNA which contributes 1% of your total DNA. Very, 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 very important. 1% DNA is your mitochondrial DNA. You should not miss it. Mitochondrial DNA contributes for about 1%. Okay. Then you have a transcription. Initiation is done by RNA polymerase, which binds with the promoter region, and elongation is done by nucleotide addition. And termination, it is row dependent or row independent. Row dependent means row cut the RNA at the termination site, while row independent is hairpin structure formation. While the row independent is hairpin structure formation and cutting off. What are the post-transcriptional modification? 5' capping, 3' tailing, and splicing. 5' capping, 3' tailing, and splicing. And apart from the translation, so activation is done by combining of amino acid with the tRNA on the amino acid arm, and then it will synthetize enzyme will combine with the amino acid and with the tRNA and converts that into amino acyl tRNA. And initiation is done by initiator codon AUG, which codes for methionine. So for in prokaryotes, it is always the Shine-Dalgarno sequence. For prokaryotes, the initiation of translation is done by Shine-Dalgarno sequence. Very, 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 very important MCQ. And the transfer of amino acid from N terminus to C terminus will happen in the initiation. Then you have a termination where you see the release of polypeptide chain after meeting the stop codon. So the released polypeptide chain will undergo post-transcriptional modification by undergoing a folding, and protein folding. Post-translational modification is so called as protein folding, and the protein folding is mediated by the protein called as chaperone protein. Very, 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 very important MCQ. Another important MCQ. Next, we are having a blotting technique. So I used to remember with the snowdrop, but there are certain people used to say the north and south stories. Whatever you want to remember. I follow the snowdrop mnemonic of your first aid. So S stands for southern blot for DNA, northern blot for RNA, and western blot for protein. Western blot for protein. Apart from that, you have a bile acid, synthesis of bile acid. So bile acid synthesis, cholesterol will be converted into 7-hydroxycholesterol mediated by 7-alpha hydroxylase, which is a rate limiting enzyme. And this 7-hydroxycholesterol will be converted into cholic acid and quinodeoxycholic acid. Okay, 7-hydroxycholesterol will be converted into cholic acid and quinodeoxycholic acid. Along with that, conjugation with the glycine and taurine, and it will form cholic acid will become glycocholic acid, and taurine will combine with the cholic acid and form a taurocholic acid. Glycocholic and taurocholic acid. And similarly, quinodeoxycholic acid will combine with glycine and taurine to form glycoquinodeoxycholic acid and tauroquinodeoxycholic acid. Glycoquinodeoxycholic acid and tauroquinodeoxycholic acid. They combine to form lithocholic acid, while glycocholic and taurocholic combine to form a deoxycholic acid. They combine to form a deoxycholic acid. Both are the bile acid. Both are the bile acids, people. So this is all about the bile acid synthesis. Then you have a test for amino acid. Ninhydrin test is a test for amino acid. Biuret is for protein. Sulfur is for methionine. Sagakuchi is for arginine. Polys is for histidine, millions is for tyrosine, aldehyde is for tryptophan, and xanthoprotein acid is for aromatic amino acid like PTT, phenylalanine, tyrosine, and tryptophan. So with this, we have completed the essentials. There are certain important images. This is the image of your tRNA, and this is the image of your lac operon gene, and this is a Watson and Crick model of DNA, primary structure, alpha helic and beta plated sheet images, and you have the 4 Ds of pellagra, that is a Casal's necklace. Here, this is the Casal's necklace, or the important image-based question that you may see in the biochemistry has been listed here. Here is a Carl Alexander Newberg, which is the father of biochemistry, where Claude Bernard is the father of your physiology, and Andreas Vesalius is the father of your modern anatomy. So all of them you have to remember. All of them you have to remember. So these are the important things we discussed about your biochemistry, people. So now we are going to do the grand revision of your BAP session. Now we are going to do the grand revision of your BAP session. So that keep the PDF next to you. Keep the PDF next to you and read the PDF and watch this revision series after that, so that you can able to remember most of the content. You can able to remember most of the content. So let's move on to the first subject, which is your anatomy. So speaking about the anatomy. What are the things you have to remember in the anatomy? First one is the upper limb, bones of upper limb. So this is going to be a Q&A session where I give you 5 seconds to give you the answer. If not, I will give you the answer. Okay. So far, speaking about the anatomy, the first area is your upper limb. So upper limb, the most common area we are going to see here is your the initiation of the topic, that is your bones of your upper limb, that is your clavicle. Speaking about the clavicle is the only long bone which lies horizontally, and the clavicle is the only long bone which intramembranous ossification, and fall on the weakest point of the clavicle is junction between medial 2/3 and lateral 1/3. Junction between medial 2/3 and lateral 1/3. Okay. Apart from that, we are having this what do we call it? We are having the scapula. So acromion process of the scapula. Acromion process of the scapula gives attachment to your deltoid fibers, while coracoid process gives the attachment to your pectoralis minor, coracobrachialis, and a short head of biceps. Then you have a humerus. Weakest point of humerus is your surgical neck. Fracture on the surgical neck could result in axillary nerve. Shaft could result in radial nerve. Medial coepicondyle could result in ulnar nerve. Supracondylar fracture could result in anterior intraosseous nerve. Anterior intraosseous nerve, which we had given and written here. Okay, then you have a radius Colles fracture, fracture of the radius with the posterior displacement. While Smith fracture, reversal of Colles fracture. Nurse-Meyer elbow, subluxation of radial head. Galeazzi fracture, distal radio-ulnar joint dislocation with the fracture of lower radius. And you have this student elbow, inflammation of olecranon bursa. And scaphoid is the bone which is most commonly fractured carpal bone. Lunate is the bone which is the most commonly dislocated bone. First to ossify is your capitate. Last to ossify is your pisiform. Metacarpals are 5 in number. First metacarpal base fracture could result in Bennett fracture. Fifth metacarpal neck could result in Boxer's fracture. And apart from that, you have this rotator cuff muscle, sits minor. Shoulder abduction 0 to 15 is supraspinatus. 15 to 90 is deltoid. More than 90 is serratus anterior. Okay, absence of sternal portion of your pectoralis major and minor is seen in Poland syndrome. Okay, then brachial plexus, nerve root of dorsal scapular nerve, C5, nerve root of long thoracic nerve, C5, C6, C7, nerve to subclavius, C5 and C6, suprascapular, C5, C6, upper trunk injury, Erb's palsy, policeman tip deformity, lower trunk injury, Klumpke's palsy, loss of function of your wrist and hand. And then you have a suprascapular nerve, C5, C6, lateral pectoral nerve, C5, C6, C7, musculocutaneous nerve, C5, C6, C7, and lateral median nerve, C6 to T1, C6 to T1, and then you have this ulnar nerve, C8, T1, and then you have this radial nerve C5 to T1, axillary nerve C5 and C6, and you have this medial brachial cutaneous nerve, which is your T1, medial cutaneous nerve of your forearm, which is your C8 and T1, upper subscapular nerve C5, C6, lower and middle subscapular nerve or thoracodorsal nerve C6, C7, C8, C6, C7, C8. Long thoracic nerve injury could result in winging of scapula. Axillary nerve, initiation of abduction will be lost. Abduction of arm will be lost. Radial nerve, wrist drop will be there. Median nerve, ape hand at the hand. And wrist, you may see cannot able to make a O with the thumb and the index finger. Ulnar nerve, claw hand. Ulnar nerve at wrist, cannot able to hold the paper between your middle and ring finger. Okay. And then you have the break, and then you have the lower limb. The anatomical location or vertebral level of anterior superior iliac spine is S1, posterior superior iliac spine is S2, iliac crest is L4, Chanting's line is superior pubic ramus to medial margin of femoral neck, Nelaton's line is anterior superior iliac spine to ischial tuberosity, pectineal line is pubic tubercle to iliopubic eminence. Femur, angle of femur is 125 in adult, 160 in children. Okay, and then patella, largest sesamoid bone formed from the quadriceps femoris muscle tendon, and tibia, most common site for your osteomyelitis, and then fibula, PART fracture is the lower end of fibula fracture, most common location of fracture is your fibular neck. Okay, and apart from that, we have the metatarsal, march fracture is metatarsal bone fracture is your march fracture, and then you have this nerve injuries, femoral nerve, loss of knee jerk reflex, obturator nerve, adduction of thigh is lost, superior gluteal nerve, waddling gait, positive Trendelenburg sign. Inferior gluteal nerve, patient will lean the body trunk backward at the heel strike. Common peroneal nerve, foot drop. Tibial nerve at popliteal fossa, dorsiflexed and everted foot will be there. Okay, anterior compartment is supplied by femoral nerve, medial compartment is supplied by obturator nerve, and posterior compartment of your thigh is supplied by your sciatic nerve. Okay, then we talk about the type of knee joint. Knee joint is a hinge joint. Extension of knee is done by ACL. Flexion is done by PCL. Locking is done by quadriceps femoris. Unlocking is done by popliteus. While nutrient artery for femur, profunda femori, tibia, posterior tibial artery, fibula, peroneal artery, and humerus, profunda brachiae artery. Profunda brachiae artery. And out of that thorax, so the imaginary plane passes between your sternal angle to T4 is junction between superior and inferior mediastinum, beginning and ending of arch of aorta, brachial bifurcation in cadaver, SVC passes fibrous pericardium. SVC passes PSS fibrous pericardium. Then you have an inspiratory muscle, which is a leading muscle or a diaphragm and external intercostal, leading expiratory or elastic recoiling of alveoli and thoracic wall. Okay, nerve supply of your thorax is thoracic spinal nerve from T1 to T2. Okay, T1 to T12, sorry. And apart from that, cardiac dominance is determined by posterior interventricular artery. If it arises from the RCA, right cardiac dominance, LCA, left cardiac dominance. IVC opening on diaphragm at the vertebral level of T8, esophageal is T10, aortic opening is T12. Right lung has a 3 lobes and 2 fissures, left lung has a 2 lobe and 1 fissure. And lingula is seen in left lung, and cardiac notch is seen in the left lung. Okay. Then apart from that, you have the carpus fascia, superficial layer of your anterior abdominal wall. Scarpa fascia is your deep membranous layer of your anterior abdominal wall. Colles fascia is a deep fascia of perineum, while Gerotta fascia is anterior layer of perirenal, and Buck's fascia is a superficial fascia of your penis. Zucker candle is a posterior perirenal fascia, and fascia of Denon-Villiers is rectovesical spectrum in the male, and rectoprostate in the rectovesical septum in a female, and rectoprostate in the male. Then you have a contents of inguinal canal, spermatic cord in male, round ligament of uterus in female. Apart from that, ilioinguinal nerve. Branches of celiac trunk, common hepatic artery, splenic artery, and left gastric artery. The main ones are common hepatic artery, splenic artery, and left gastric artery. Okay. Apart from that, you have this the circle of Willis formed by vertebral artery and internal carotid artery, and then pharyngeal arch. Pharyngeal arch 1 derivatives are multiple M's makes the sky slow and volatile. Malleus, incus, malleus, incus, sphenomandibular ligament, maxilla, mandible, zygomatic, temporal, sphenoid, and vomer bone. While pharyngeal arch 2 are 5S, tape, styloid, stylohyoid ligament, small horn of hyoid, and superior surface of your hyoid. One, apart from that, you have the remnant cartilage of your larynx come from pharyngeal arch 6. Muscles of mastication 1, muscles of facial expression 2, stylopharyngeus is 3, muscles of pharynx is 4, and muscles of larynx is 6. Nerve, mandibular nerve for 1, facial nerve for 2, glossopharyngeal nerve for 3, vagus nerve for 4, and recurrent laryngeal nerve for 6. Remember, all of the following muscles of your larynx are innervated by recurrent laryngeal nerve, except the exception is cricothyroid, which is supplied by your superior laryngeal nerve. Okay, superior laryngeal nerve. Among the lymph node attachment, para-aortic, testes, ovaries, kidneys, and uterus. Internal iliac, lower rectum above the pectinate line. Superficial inguinal, lower rectum below the pectinate line. Very, 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 very important. Okay, below the pectinate line. These are the important things you have to know in terms of your anatomy. So everything which has been mentioned here are pretty high yield, and everything you have to know, people. So don't miss it and learn everything in the anatomy. Okay, now moving on to the physiology. Speaking about the physiology, the important ones were your general physiology and cardiovascular physiology, and then neurophysiology, and then you have your GI physiology. So we're gonna move on to the general physiology. So term homeostasis was coined by Walter D. Cannon. Total body water was 60%, and deuterium is the radioactive dye we use for the measurement of total body water, and radioiodinated albumin 125 is used for the measurement of plasma, and membrane transport, active transport, primary. A classical example is sodium potassium ATPase. Secondary classical example is SGLT. Facilitated diffusion, classical example is your GLUT. Classical example is your GLUT. Among that, you have the cardiac action potential, phase 0, sodium influx, and phase 1, potassium efflux, phase 2, potassium efflux and calcium influx, phase 3, potassium efflux, and phase 4, sodium potassium ATPase. While SA node and AV node, phase 0 depends on L-type calcium channel. Chronotrophy, heart rate. Ionotrophy, myocardial contractility. Chromotrophy, cardiac conduction velocity. Bathmotrophy, cardiac excitability. Lucitrophy, cardiac relaxation. Okay. Then apart from that, you have a cardiac cycle. S1, isovolumetric contraction. S2, protodiastole. S3, rapid filling. S4, atrial contraction. Then we are having this cardiac output formula, heart rate into stroke volume. Stroke volume depends on 3 factors. What are they? Preload, afterload, myocardial contractility. Preload, afterload, and myocardial contractility. Okay, among that, afterload is inversely proportional to cardiac output. Among that, afterload is inversely proportional to cardiac output. What is the normal mean arterial pressure? 93.33 mm of Hg. 93.33 mm of Hg. And apart from that, what is the hormones which has been synthesized, or the substances which has been synthesized from what cell? G cells, gastrin, gastric acid production, growth of mucosa of your GI tract. I cells, cholecystokinin. S cells, secretin. Kappa cells, GAP. D cells, somatostatin. Mo cells, motilin. And growth hormone, ghrelin. Carbohydrate, location of absorption is duodenum. Lipids, duodenum and jejunum. Protein, duodenum and jejunum. Vitamin B12, terminal ileum. Iron, duodenum. Iron, it is a duodenum. Apart from that, lung volumes. So total lung capacity is all 4 combination: ERV, IRV, RV, and TV. And normal FEV1 FVC ratio is 80%. As per the Pinali's law, decrease less than 80% indicate obstructive lung disease. And mechanics of breathing, we already discussed in the anatomy. Resistance, major situs, medium sized bronchi. R is equal to 8 rho L divided by pi R power 4, where rho is viscosity, L is length, and R is the radius. Okay. A ventilation perfusion ratio, it should be 0.8. V is equal to 0, ventilation defect. V is equal to infinity, perfusion defect. Okay. And apart from that, factors which can able to shift the oxygen dissociation curve to right. What are they? Increase in PCO2, decrease in pH, increase in temperature, high altitude, during exercise, increase in 2,3 dpg. Increase in 2,3 dpg. Apart from that, you have a normal DLCO is 25 ml per minute per mm of Hg. Herring-Breuer inflation reflexes. Overinflation prevent further lung inflation, and Herring-Breuer deflation reflex indicate deflation increases ventilatory rate, and paradoxical reflex of your head indicates inflation of lung causes further inflation. Okay, apart from that, we are talking about the endocrine physiology. So growth hormone, somatostatin's production, decrease in glucose uptake and lipolysis, and increase in protein synthesis. Prolactin, milk production, breast development, suppression of ovulation, and suppression of spermatogenesis. Posterior pituitary hormone, ADH, stimulation of aquaporins, oxytocin, milk ejection, and contraction of uterus. Thyroid hormones, main function is your maturation of CNS, increase in the PMR, and increases the oxygen consumption, and increases synthesis of sodium potassium ATPase, increases cardiac output, and increases glucose absorption. And adrenal cortex, zona glomerulosa secretes aldosterone, fasciculata secretes cortisol, reticularis secretes androgens. Among that, Addison's increased ACTH, Cushing's decreased ACTH, and you have the Cushing's syndrome, decreased ACTH, and Cushing's disease, increased ACTH. Cone syndrome, aldosterone tumor. Cone syndrome is aldosterone tumor. Apart from that, endocrine pancreas was there. Alpha cells glucagon, beta cells insulin, delta cells somatostatin. Then Sertoli cell secretes inhibin, Leydig cell secretes testosterone. Lactation, estrogen and progesterone, growth of breast. And prolactin, milk synthesis. Oxytocin, milk ejection. Oxytocin, milk ejection. Major excitatory neurotransmitter in CNS, glutamate. PNS, acetylcholine. Influx of sodium ions in the excitatory. Influx of chloride ions in the inhibitory. Fastest conducting fiber is your A alpha. Slowest is your type C. Type C is most susceptible to local anesthesia. Type B is most susceptible to hypoxia. Type A is most susceptible to pressure. Type A is most susceptible to pressure. Apart from that, structural protein, titin, desmin, actin, and dystrophin. Titin is for elasticity. Desmin is for desmin connects to Z line. Actin binds the actin with the Z line. Dystrophin will add the strength to the muscle. Then you have a regulatory protein, troponin C, I, and T. C for calcium binds with troponin C, and I for actin myosin interaction. T for troponin components to tropomyosin. And then calsequestrant, which is a calcium buffer. Okay, which is a calcium buffer. Apart from that, what are the important circulation of the CSF? CSF production is 500 to 700 ml per day. Circulating CSF volume is 150 ml. Circulating CSF pressure is 300 to 600 mm of water. And the pathway is lateral ventricle to foramen of Monro, foramen of Monro to third ventricle to cerebral aqueduct of Sylvius, Sylvius to fourth ventricle to foramen of Luschka and Magendie, and then to the subarachnoid space, So flexion is mediated by rubro, the extension is mediated by vestibulo, and respiration muscle tone is mediated by reticulo, and auditory impulse is mediated by tectospinal tract, and your pain, temperature, crude touch is maintained by spinothalamic. Vibration, proprioception, 2-point discrimination is done by your dorsal column. With this, we are finishing your physiology. So we have covered biochemistry, anatomy, and physiology revision, people. We have covered all the three. So today we pass our session. Tomorrow we will do anesthesia and dermatology. So we will make you relax for one day, so that we do the short subject, so that you get enough break to cover up your BAP. After that one day to finish up your remaining portion, we do our second year subject like pathology, microbiology, and pharmacology. Okay. So signing off to you from for today, and have a good read, and all the very best for you people. Tomorrow, see you again with the anesthesia and dermatology.